everyone. If you're new here, I'm İnci. I'm a third year molecular biology and genetics student at the Acıbadem University with a GPA that you can say pretty fair, 3.45, which basically means I spend more time with my textbooks and micropipettes than with actual humans. I started İnci anlatıyor because I believe science shouldn't stay locked inside cold laboratories or dense academic papers. My mission is to be the bridge between complex lab data and you. Whether you're a fellow student trying to survive finals or a curious mind wondering how life works at the atomic level, you're in the right place. Let's decode the world together. Today we are starting a journey into the heart of biology. I want you to think about a tiny microscopic cell. Inside that cell's nucleus, there's a string of DNA that is 2 meters long. If you took all the DNA from your body and stretched it out, it would reach the sun and back multiple times. How does that much information fit into space you can't even see? And how does it stay so organized that your body knows exactly how to build a heart cell versus a brain cell? Today we are cracking open the master archive, DNA. The basic anatomy, the twisted ladder. Most of you know DNA looks like a twisted ladder, the double helix. But as MBG students, we need to look closer. This ladder has rails and rungs. The rails are made of a sugar phosphate backbone. It's like the steel frame of a skyscraper. But the magic happens in the rungs, the nitrogenous bases, adenine, thymine, guanine, and cytosine. These are the letters of the genetic alphabet. Everything that just makes you, is written with these 4 letters. Okay, now it's time for some hard science for my fellow students preparing for their exams. Let's talk about why DNA is the ultimate storage molecule. Stability of the bonds. 2 strands aren't just floating, they're held together by hydrogen bonds. But they aren't all equal. A pairs with T with 2 hydrogen bonds. G pairs with C with 3 hydrogen bonds. Quick exam tip. This is why GC-rich DNA is much more stable and has a higher melting temperature, Tm. If you are studying an organism that lives in a hot spring, expect a lot of GC pairs. The antiparallel geometry. DNA is antiparallel. Think of it like a 2-way street where cars drive in the opposite directions. One strand runs 5' to 3', and the other one runs to 3' to 5'. Why does this matter? Because every enzyme in your body, like DNA polymerase, is a one-way machine. They can only read or write in 5' to 3' direction. This orientation determines how we replicate and how we live. Growth and protein handshake. Look closely at the helix. It's not perfectly symmetrical. It has a major groove and a minor groove. Most proteins that read your DNA, like the transcription factors, prefer to bind the major groove because it has more chemical information exposed. It's like the difference between reading a book through a wide open window versus a tiny keyhole. Now remember that 2-meter string. How do we fit in it? We use proteins called histones. DNA wraps around these histones like thread around a spool, forming nucleosomes. This isn't just for space, it's for control. When DNA is highly packed, heterochromatin, it's silent. When it's loose, euchromatin, it's ready to be read. It's the ultimate molecular origami. And final question of this part. Why do we use DNA for storage and not RNA? And the chemistry has the answer. DNA uses deoxyribose, which lacks one oxygen atom compared to RNA's ribose. That missing oxygen makes DNA much less reactive. Also, DNA uses thymine instead of uracil. This is a genius move by evolution because it makes it easier for repair enzymes to spot errors. DNA is built for permanence and RNA is built for action. So, we have the master archive. It is stable, it's organized, and it's packed with information. But a book that stays on the shelf, it's useless. How do we take these instructions and turn them into a message that rest of the cell can understand? In the next part, we are going to look at the transcription, how we make a molecular photocopy of the master script. And I will see you in the part 2. Hello everyone! In the part 1, we talked about DNA being the master archive. But here's the problem. DNA lives inside the nucleus, and it is too precious to leave. However, the machines that build our body, the ribosomes, are outside in the cytoplasm. So, how to get instructions from vault to the factory? We don't take the original book. We make a photocopy. This process is called transcription, and the copy we make is mRNA, messenger RNA. Transcription doesn't just start anywhere. Imagine a 2-meter long string. You need to find the exact start of a gene. For my curious minds, the enzyme RNA Polymerase looks for a specific sequence called promoter. In eukaryotes, we often look for the Tata box. Once the polymerase binds there, with the help of transcription factors, it unzips the DNA and the magic starts. And this is where the most students lose points in the exams. So listen closely. DNA is double-stranded. RNA polymerase only reads one of them. The strand being read is the template strand, 3' to 5', but the resulting mRNA looks exactly like the other strand, the coding strand, except it has uracil instead of thymine. Why? Because life is a mirror image. The polymerase writes in 5' to 3' direction, watching the code of the master script perfectly. In bacteria, the mRNA is ready immediately, but in complex organisms like us, the initial script pre-mRNA is a mess. It's full of junk sequences called introns. We use a molecular machine called the spliceosome to cut out the introns and glue the exons together. Think of it like a film editing. We cut out the bloopers and keep the action scenes. Before the mRNA can leave the nucleus, it needs 2 protective suitcases, 5' cape, a modified guanine head that helps the ribosome recognize it, and a poly A tail. A long string of adenines at the 3' end of mRNA that protects the script from being eaten by the enzymes in the cytoplasm. It's a countdown timer for the mRNA's life. So, our photocopy is made, edited, and packed. It's leaving the nucleus and heading the molecular factory. But how do we turn a sequence of letters into a physical 3D protein that can actually do something? In part 3, we are going to look at translation, the most incredible language translation in the universe. And I'll see you in the ribosome. Stay curious for part 3. Hello, I'm back again. In part 2, we saw how DNA is photocopied into the mRNA. But now, we face the ultimate biological challenge, language translation. Our cell needs to translate the language of nucleotides into the language of amino acids. This is translation, and arguably the most complex 3D printing process in the known universe. Before the printing starts, we need the raw materials. You've heard of tRNA, the adapter molecule. But for my science lovers, Here is a real hero, amino acid tRNA synthetase. This enzyme is the molecular dictionary. It ensures that the correct amino acid is attached to the correct tRNA. If this enzyme makes a mistake, the entire protein is ruined. It's the ultimate quality control officer of the cell. But how do four letters A, U, G, C code for 20 different amino acids? Through codons, groups of 3 letters. If we take into consideration that for 3 codons, every possible space has 4 options of the bases. So 4 times 4 times 4 equals to 64. So there are 64 possible codons. But wait, we don't have 64 different tRNAs. Why? Because of the wobble hypothesis. The first 2 positions of a codon require strict base pairing, but the third position is flexible or wobbly. This allows one tRNA to recognize multiple codons. It's nature's way of being incredibly efficient with its resources. And now we are ready to enter the factory, the ribosome. It's made of 2 subunits, 60S and 40S in humans. Inside, there are 3 specific rooms where the action happens. A site, amino acid. This is the landing strip where the new tRNA arrives. P site, peptidyl. This is the construction site where the growing protein chain is held. The enzyme peptidyl transferase, which is actually a part of RNA, forms the peptide bonds. E site, exit. This is the departure gate where the empty tRNA leaves to be recharged. It all starts with the start codon, AUG, which always calls for methionine. The ribosome zips along mRNA, adding amino acids one by one until it hits a stop codon, UAA, UAG, or UGA. Crucial detail: there are no tRNAs for stop codons. Instead, release factors enter the ribosome and literally spark a molecular explosion that releases the final finished polypeptide chain into the cell. But just a long chain of amino acids isn't a functional protein yet. It's need to fold, twist, and turn into perfect 3D shape. In the final part of our series, we'll see how cells control this entire process and what happens when software bugs or mutations try to ruin the script. I'll see you in the part 4. So, we reached the final of our Central Dogma series. We know how DNA becomes protein, but there's the biggest question in biology. If every cell in your body has the exact same DNA, how does a neuron look like a long wire while a muscle cell looks like a sturdy fiber? The answer isn't the code itself, it's the resolution. It's not about what you have, it's about what you use. The dimer switches transcription factors. Think of your genes as light bulbs. Transcription factors are the people standing at the switches. Activators turn the lights up, helping RNA polymerase to find the gene. Repressors block the switches, keeping the gene silent. In humans, this is a high-stakes dance involving enhancers and silencers that can be thousands of base pairs away from the gene that they control. Epigenetics, the sticky note system. Now let's talk about the hottest topic in modern biology, epigenetics. This is control beyond the DNA sequence. Imagine your DNA is a massive encyclopedia. DNA methylation. This is like putting a Do not read sticky note over a paragraph. It shuts down the gene. Histone modification. Remember those histone spools DNA wraps around? If we add an acetyl group, the DNA loosens up euchromatin and it says read me. If we add a methyl group, it's packed tightly heterochromatin and goes to sleep. This is how your environment, your diet, and even your stress levels can influence your gene expression without changing the single letter of your DNA. But what happens if these letters do change? So look at different types of mutations. That's a mutation. Point mutations. One letter is swapped, silent. No change in the protein thanks to the degenerate code. Missense, a premature stop codon is created. This is like a movie ending after 10 minutes is started. And frame shift mutations, adding or deleting a letter. This shifts the entire reading frame. Every single word after the error becomes gibberish. These are almost always devastating for our cell. So, from the rock-solid archive of DNA to the dynamic markers of we call proteins. The Central Dogma is the story of you. It's a story of incredible precision, but also of beautiful flexibility. Understanding this flow is the key to curing diseases, engineering new medicines, and appreciating the sheer miracle of being alive. Thank you for joining me on this deep dive. I'm heading back to my studies now, and stay scientific, keep asking why, and I'll see you in the next masterclass. And all the topics title that I mentioned in this video will be in the description box down below. Don't forget to subscribe and hit the thumbs up button. See you in the next one. Bye.